As scientists, we're often taught to think of things in binaries: treated versus control, disease versus normal. We love that our scientific method can help us to find truth in a complex universe. But underneath all of these statistics and controlled trials is a messy, nuanced reality. As a society, we've begun to talk more about spectrums of behaviors, treatment approaches, and diagnoses. But as scientists, we sometimes trail the pack in these conversations. There's perhaps no better example of this than autism, known as Autism Spectrum Disorder, or ASD, which is a highly diverse neurodevelopmental disorder, both in terms of presentation, meaning what we can observe from behaviors, and genetic causes. Traditional methods for studying the causes of brain disorders rely on two broad approaches. Forward genetic approaches can help us to causally link genes with a disorder, such as autism. Backwards approaches, on the other hand, rely on molecular interrogation, usually from postmortem brain. This gives us a snapshot in time, a sort of state of the brain, if you will. And while many neurological disorders are diagnosed based on this snapshot in time, think Alzheimer's, for example, psychiatric disorders are classically thought of as lacking a visible pathology. And so, somewhat surprisingly, especially given the wide range of human experiences of autism, a consistent molecular signature has been found in a majority of cases. What this means is that, on some level, if we look at the molecules within cells, we can almost see autism in the brain. This signature is defined by a decrease in synapse-related genes and markers of inhibitory neuron activity, and an increase in neural immune activity in glial subtypes of the brain. However, this alone is not sufficient to decipher cause and effect, forcing us to act as sort of neural archaeologists, looking back in time from the end result to try to understand what might have happened during development, when might it have happened, and where in the brain might these changes be taking place. Now, forward approaches can guide us to a certain extent. We know, for example, genes causally tied to autism are enriched in processes such as neurogenesis, the birth of new neurons, and corticogenesis, the formation of the cortex. And not surprisingly, perhaps, these genes are most highly expressed during fetal brain development. But how can we link hundreds of genes causally associated with autism through developmental pathways to this adult molecular signature? Well, if we think of each one of these genes as its own sort of tributary, starting at the top of a mountain and flowing downhill into a river in the valley, we can imagine the challenges of tackling each one of these in a one-by-one fashion atop their own mountain. But if we look to see where these tributaries converge at the level of the river, we can see the importance of studying shared biological properties that are happening across genetic causes. And if we think of the water flowing downhill as the passage of time, we can see how important it is to understand when this convergence is taking place in order to form effective treatment strategies to intervene. So in order to bridge this gap between diverse genetic causes and molecular signatures, science really needs new techniques to be able to see in the middle. Luckily for us, we've recently been granted a major breakthrough: the organoid model. Organoids are made by getting cells from patients, usually in the form of a skin biopsy, and then driving those cells into stem cells, and then down a path of differentiation to model brain development. Organoids are a game changer in our ability to look at diversity within a diagnosis, because we can get cells and make organoids from a range of patients with different genetic causes. They're also excellent for our ability to look at development, because within a particular individual, we can make multiple different organoids and then sample those across different points of development. In this way, we can help to connect genetic causes with molecular signatures through understanding the different pathways that are taken over development. In my project, I'll examine 5 high-confidence autism risk genes in 3 different genetic backgrounds at 2 specific time points chosen to encompass the peak of neurogenesis. We've intentionally selected genes that represent a range of biological properties associated with autism, everything from chromatin remodelers to synaptic organizers. This will help us to connect the dots from genetic causes to molecular signatures through time. But another way that we can better look at the passage of time is to become even better archaeologists of the adult brain. To do this, we'll use cutting-edge bioinformatic network analysis tools to derive transcription factor regulatory networks in the adult brain. In this way, we can look at the relationship between a transcription factor and its downstream target genes and understand how those genes causally linked with autism may regulate a large amount of the changes seen in the molecular signature. We can then make predictions about how to intervene to change the transcription of many genes by leveraging their relationship to a shared driver. Finally, we need to address the where question. Where in the brain are these changes taking place? To do this, we'll use spatial transcriptomics, another novel technique that allows us to actually look and see what specific cells are these changed transcriptional regulatory networks happening within. Are they in particular circuits that connect specific cells to each other? Maybe they're concentrated in specific layers of the brain or within cortical hotspots, say. Broadly, this project will examine convergent changes in autism. When do those changes happen, and where in the brain do they become instantiated? Genetics are a powerful force to reckon with. And in psychiatry, we walk a fine line in trying to both uplift the diversity of the human experience and also provide tools to treat when that diversity makes it challenging to live in the world. I know this is a complex task, but I do believe that as scientists, we're up for the challenge.